today is all about the alternative methods for carbon fixation or alternatives to photosynthesis. This is a relatively shorter lesson because I've got an exit ticket for you and a little bit of time to get together to finalize and submit those lab reports, which need to be in by tomorrow. So, starting us off, my goal is for you to be able to differentiate between C3, C4, and CAM plants. Those are things we are going to talk about today, and explain their evolutionary advantages. So, we're going to start off with one of those AP biology big ideas being our evolution connection. So we know about stomata, those little holes in a leaf tissue in order to release oxygen and take in carbon dioxide. We've talked about those. We've interacted with them in our lab. What we haven't really talked about is this is where transpiration occurs. Transpiration is a relatively big part of the water cycle. It is the evaporation of water out of plant tissue. So, water, when it is in plant tissue, can be evaporated out by the heat of the environment. So, our plants, in particularly hot, dry environments, have to balance photosynthesis and water loss. They have to They have to make sure that the stomata are allowing the good stuff in and the bad stuff out without sucking all of the water out of them, because they need water to do that first step in the light reactions, right? So we have to balance this photosynthesis and water loss. And sometimes what happens in order to kind of maintain this, or as a result of this, when carbon dioxide is too low, so there's not any carbon dioxide getting in, and there is too much oxygen building up in our leaf cells, because we have either closed the stomata to kind of stop the hemorrhage of water, or because the cell just is really, really high on carbon dioxide and really low in carbon dioxide and high in oxygen. They go through a process called photorespiration. Photorespiration. This process is really wasteful. This process doesn't do much for the overall health of the cell. In fact, we don't know exactly why plants do this, but there is a kind of a second process called photorespiration. I guess it's a third process when we take in cell respiration into account. But this process can be considered a evolutionary leftover, or a couple other things that we'll talk about in a moment. But this is what happens when plants are in a really, really hot space, and water is being lost faster than they can take it up. So plants have to adapt in order to make sure they have everything they need to carry out photosynthesis. So this is our big evolution connection for our photosynthesis unit, or segment of the unit. So, when carbon dioxide is low, so there's not enough carbon dioxide to feed into the Calvin cycle, oxygen is high, so oxygen has built up in the cell, our plants will go through a process called photorespiration. And all plants can do this. It's just what plants do to avoid doing this. So I clicked on a thing and now my clicker's not working. All right, so what is photorespiration? Why is it wasteful? Photorespiration uses light, consumes oxygen, and releases CO2 in order to build that CO2 concentration back up. This process uses ATP and decreases the photosynthetic output. It decreases how much sugars are made through photosynthesis. This process is costly, and we do not understand the point of it. Some people think that it is an evolutionary leftover or evolutionary baggage, something that other plants had done, ancestral plants had done in the past that helped them, but like the chemical process hasn't like been taken out of the DNA of current plants. Some think that it is a leftover use of Rubisco. So Rubisco is doing multiple things in the cell before it found its real role in the Calvin cycle. Others think it provides protection by using that oxygen buildup before the oxygen starts causing damage in the cell, because oxygen can cause damage in a cell. So some people think it's just like damage control, like get the oxygen concentrations down so stuff doesn't go awry. But the main idea of it is that it uses light, so photo, and consumes oxygen, respiration, right? It releases that CO2. So cell respiration, whenever we were talking about cell respiration way back, it seems like eons ago, we consumed oxygen and released CO2. And I say we as in we, because we do cell respiration. Plants do it too, but cell respiration. So, but we're using light, so we call it photo respiration. It is counterproductive and rather pointless. So, as you explore biology, you kind of figure out more and more that these processes aren't perfect. They're not efficient. And evolution doesn't always push things to the best, most ideal scenarios. Sometimes we are left with weird little processes that have little to no efficiency or point, just because that's what happened. So, nothing's perfect, not even photosynthesis. So, we've got this kind of weird side chain photorespiration. And there are two alternative modes for carbon fixation. So our plant species, some of them have alternative processes or like add-ons to photosynthesis to minimize photorespiration. This keeps the cell from doing those wasteful processes. I don't know what this enter did in there. So there are two types of plants that have evolved over time to minimize this photorespiration, to minimize this wasteful process. They are called C4 plants and CAM plants. Now, the plants that we've been talking about, like the sunflowers and daisies that we've been talking about, that do regular old photosynthesis, we call that C3, and you'll learn the difference between C3 and C4 momentarily. But C4 and CAM plants are our two alternative ways to get carbon dioxide out of the atmosphere and into carbohydrates. So, the whole goal of this is to stop the plants from doing that wasteful process of photorespiration. How do we avoid losing our photosynthetic output? I say we, I mean plants. Specifically, C4 and CAM plants. So, starting with C4 plants. In C4 plants, these are like sugarcane and corn and big like thicker grasses. These are called C4 plants. These are examples of C4 plants. And what happens is before they get to the Calvin cycle, there's a different form, a different way to get carbon out of the atmosphere that forms a 4-carbon compound at its first, as its first product. So in C3 plants, right, we talked about that 3GP being our product, right? At the very end of the Calvin cycle, hi, thank you. At the very end of the Calvin cycle, we had that 3GP molecule, and those were that was our 3-carbon, right? Because 3GP. So in C4 plants, we have a 4-carbon for C4, whereas our 3C was our 3GP. So this process of using carbon fixation that starts with a 4-carbon compound that is then fed into the Calvin cycle is believed to have evolved independently at least 45 times. Which means if we had we have a common ancestor, right? A common photosynthetic ancestor. This is what it means to have evolved independently. We've got our common ancestor. It means as this branched off evolutionarily, and we'll talk about cladograms when we talk about evolution, that means that this C4 process, this alternative carbon fixation, popped up on its own. Meaning it wasn't down here, like a common ancestor didn't have it and pass it down to all of these different types of C4 plants. These C4 plants came up with it on their own. At least 45 different plant species came up with this process, or evolved to have this process. This idea of different species evolving the same thing independently is not uncommon. The crab-shaped body type has evolved independently on its own hundreds of times. Evolution really likes crabs, apparently, and we'll talk about that when we get to evolution towards the end of the year. But C4 is another example of how things just kind of pop up on their own. So, the anatomy of these plants is really interesting because that's our big difference. That's where we get, this is how we save that water loss to prevent that photorespiration. And we do this because of the plant's anatomy, the shape and type of cells that these plants have. So they've got the mesophyll cells, those mesophyll cells that we've been talking about all through photosynthesis. But they also have a second type of cell called the bundle sheath cells. And these bundle sheath cells are exactly what they are. They're bundled up together and they're sheathed really tightly into veins on the leaf. So we can kind of see, if we looked at corn plants or sugarcane, we can see these little bundles right here. So this is down here a picture of a plant cell, and we've got our bundle sheath cells right through here, and we've got our mesophyll cells all around the outside of them. The way these cells mitigate water loss is by taking the Calvin cycle and only doing the Calvin cycle in these bundle sheath cells. So we incorporate carbon dioxide in the mesophyll cells, and we kind of compartmentalize. If we compartmentalize, we don't have that buildup of carbon dioxide in all the cells all at once. We can keep our processes separate and just kind of assembly line it through these different types of cells. This kind of prevents the buildup, the increases in concentration of carbon dioxide and oxygen in one type of cell, and we can just kind of keep it going by sending it on to the next kind of cell. So Our bundle sheath cells are really tightly packed into our plant tissue, and they are where the Calvin cycle happens. The light reactions still happen in our mesophyll, but they send everything into the bundle sheath cells to get stuff done. And we're not going to get super into detail like we did with photosynthesis with like step-by-step process. I'm going to show it to you, but we're just going to hit the big ideas in it before moving on. So I'll get my handy highlighter out, and you only have to write down what I highlight. That way we don't have to write down each and every little chemical step, but I think it's important that I tell you what it's about, in case you see a chemical pathway where you have to identify different things. Being used to seeing chemical pathways make them easier to interpret. So we've got our went backwards. Yeah. So this is our Calvin cycle in C4 plants. There are 4 big steps that happen. Yes. Okay. 4 big steps. Number 1, we've got our, so step 1, didn't highlight because of course. We've got an enzyme in the mesophyll adding carbon dioxide to a molecule called PEP. There is a special enzyme. That enzyme's name is PEP carboxylase, because it adds that carboxyl group to PEP carboxyl ACE. Remember that ACE means enzyme, and the name of the enzyme tells us what it does. So PEP carboxylase adds a carboxyl group to PEP. PEP carboxylase is important in these plants because they can fix carbon dioxide when Rubisco can't. If we've got all of our Rubisco shoved into those bundle sheath cells where the Calvin cycle is happening, we need something in the mesophyll to fix carbon dioxide. So we have PEP carboxylase. So our second step is our 4-carbon product is now going to the bundle sheath cells. So we get that C4 product to our bundle sheath cells, and they go through plasmodesmata, which, if you remember from our, cell transport, we have our cell walls, right? And we just have these little holes in our cell walls, where that's a really bad picture. But they're essentially holes in the cell wall where cell's cytoplasm is connected. So after we've got our C4 product to the bundle sheath cells, we've got our carbon dioxide release it, and then it is refixed by Rubisco. It is refixed by Rubisco. So our 4-carbon intermediate, or a 4-carbon product, is now in the bundle sheath cells, and then it will release carbon dioxide. It will be broken down into carbon dioxide, which is fixed in Rubisco, and that will go through the Calvin cycle. In this process, in C4 plants, there is no linear electron flow. It is only cyclic. It's only cyclic electron flow. So remember, cyclic electron flow was a little bit less efficient than linear electron flow. But it also means they only have one photosystem. But that is the process of essentially respiration in C4 plants. We still have the light reactions being the same, except for they only use cyclic electron flow. But we have this kind of intermediate step where we fix carbon before we get to the Calvin cycle. CAM plants, instead of isolating these processes chemically, they isolate them physically. CAM plants open their stomata at night and then close them during the day, during the heat, to conserve water. So at night, all of these plants open their stomata, allow carbon dioxide in and oxygen out. And then at night, and then in the morning when it starts getting hot again, those stomata will close. So we're conserving water while our stomata are open, so at night. Those plants will take in carbon dioxide and fix them into organic acids to store them for the daytime. So it's taking in all of the carbon dioxide they can, fixing them into acids to store them. And this process is called crassulacean acid metabolism, or we call it CAM, because CAM is a lot easier to say than crassulacean, which I'm not even sure I'm getting that right, acid metabolism. So this is CAM plants. We see this in desert environments. You get a lot of cacti, you get a lot of tropical fruits and plants. Pineapples are CAM plants. And we've got a lot of succulents. That's the word I can think of. Succulents are often CAM plants. So during the day, when it's hot, they will close their stomata. At night, they will open up their stomata, take in carbon dioxide, and fix them into organic acids. So this is nighttime for CAM plants. Open up those stomata, let the oxygen out, let the carbon dioxide in, and make some organic acids. Now, they will store the organic acids that they made at night in their vacuoles until the next morning when the stomata close. This is the next slide. It looks a lot similar. So during the day, we have light available. So we've got the light reactions going. And once we've got the light reactions going, we now have the ATP and NADPH, or the energy, for the Calvin cycle to go, because the Calvin cycle needs energy, right? But we need the light reactions to get that energy. So we stored up all the carbon we need at night, and then during the day, our Calvin cycle now has the energy from the light reactions to use up all of the carbon that it fixed at night. So it uses the nighttime to prepare for all of the photosynthesis that happens during the day. So our C4 plants kind of separated they're like compartmentalized within with like physically within the cell. And then our CAM plants isolated these processes temporally or within time. So they've got a special time for preparing and a special time for photosynthesis to happen. I've got a little diagram that'll help us kind of visualize this a little bit more. We doing okay? So, I need a couple more minutes. This is got a lot more stuff. So, nighttime, we stock up on carbon dioxide, wait till the day when we get energy. I say we, I mean CAM plants, and then we move forward. So, this diagram would be very, very helpful to kind of sum up everything that we've talked about today. So, In a C3 plants, in our regular old plants, what we do is we have carbon dioxide come into the plant, into our mesophyll, and then we have the Calvin cycle, right? We fix carbon, send it in the Calvin cycle, no big deal. And then in our C4 plants, we've got the mesophyll and the bundle sheath cells. So in our C4 plants, our carbon dioxide will come in, get fixed into our C4 or our C4 intermediate. That will go into the bundle sheath cells where it'll release carbon dioxide and go through the Calvin cycle. And then finally, our CAM plants, this says C4. I'm going to change it to organic acid so we don't get confused with that 4. That carbon dioxide comes in at night. We store our organic acids, and then during the daytime, we break that back down into CO2, and we can go through the Calvin cycle. So we can kind of see the difference here. I'm also going to pass out this chart that has a bunch of different ways that we can differentiate C3, C4, and CAM plants. Now, there's a lot of information on this chart that we did not talk about. There's a lot of information on here that we did not talk about. That does not mean that you absolutely need to know it. I just wanted to show you all of the different ways that we could differentiate. So we've got specific temperatures, we've got different enzymes, we've got our like where they're found. We've got different types of cells. We've got some examples. We've got some comparisons between the Calvin cycle, all that fun stuff. They mentioned something called the Hatch and Slack cycle. It is very similar to the, or it is an addition to the Calvin cycle that goes a little bit more in detail that we don't really need to know. But I gave you all the differences anyway. So, everybody get one? Cool. So the plan for the rest of the day, we've got a decent amount of time left, like 25 minutes. I want you to make sure you've got this diagram in your notes. I'll leave it up here on the board. You'll have an exit ticket to fill out. Nothing too crazy. I think it's only 3 questions. And then I want you to, if you've got the time, see if you are ready to turn in your lab reports, because those are due tomorrow. Anybody have any questions? All right.